As Rosemary said, my name is Adrian Fox. I'm the principal virologist at the Food and Environment Research Agency in York. We are currently a government agency. We are responsible as the National Plant Protection Organisation laboratory, but we also get involved in internal plant health issues as well, and that's how this came about. Okay. So what is carotte internal root necrosis? I think hopefully you can see virus symptoms are never easy to show on a PowerPoint presentation, but hopefully you can see this browning around the core of the carrot that extends from the crown down towards the tip. And this problem has been known about in the UK for at least 10 years. I think we're the third research group to chase it and try and find a causal agent for this problem. Initially, people thought it was a type of crown rot, but thanks to one UK horticultural consultant who was really like a dog with a bone, he wouldn't let go and he was insistent that it was a viral problem, and so we carried on chasing it down. So why is it a problem? It's internal, you can't grade it out. And over the past 10 years, the UK has seen a shift towards processing carrots and away from the fresh market. And if you get, say, more than 5% of this symptom going through a processing line, it can actually lead to a whole crop being rejected because of the unsightly brown marks. Either that or it turns up in somebody's spaghetti bolognese, and then they phone the supermarket they bought the spaghetti bolognese from, and it becomes a whole customer complaint issue. So through some previous work, which was funded by the Horticultural Development Company that Rosemary explained to you earlier, we suggested a possible loose association with virus, but we'd only looked at symptomatic individuals, and there was nothing clear coming out of that, so we wanted to look a bit more deeply. So how do you chase down an intractable symptom, a symptom where you don't actually know the cause? I thought I'd use this map of the UK because it's the last chance I may get to have a map with Scotland on it. Most of the work was actually done, though, just here around York, which is also where FERA, my institute, is based. And it just so happens that one of the UK's largest processing carrot producers is based locally with us. So it's quite good because it gives us lots of fields we can go and look at on a local basis. So we identified a stock on a processing line with necrosis symptoms. That was approximately 3% affected roots. And once we'd identified that stock, we sliced roots until we had 100 affected individuals. And then from the discard line, so all the unaffected, we pulled 100 roots out of that. So we had 100 affected, 100 unaffected, that we could then compare the two populations. As you'll see from the numbers there, it didn't quite work out like that, but that's what happens when you're trying to count carrots next to a grading line. Those carrots were then tested by conventional PCR for the viruses that we've always tested for in the UK. That's parsnip yellow fleck and the viruses of the carrot motley dwarf complex, so carrot red leaf associated viral RNA, and carrot motley dwarf virus. From that, we then took a subsample of 24 carrots, 12 affected, 12 unaffected, with a mixture of different virus symptoms. So you had some affected with virus that we'd already detected, some affected with no virus detected, and so on and so forth. And we put those through a deep sequencing route. You may have heard some explanation of deep sequencing, or next generation sequencing as it's called, through some of the genomic work that's going on. Well, we're investigating this technology for use as a tool in plant pathology, so we're using it slightly differently. And then from what came out of that, we looked at the relative incidence of the viruses in the two populations for significant differences to try and work out whether we could associate a virus or a combination of viruses with internal necrosis. So when we ran those conventional PCRs, here you've got the viruses we're looking for, so parsnip yellow fleck virus, carrot red leaf associated, and carrot mottle, and those that tested negative. And as you'll see, you've got relatively even amounts of negative, so no virus detected when you're looking for these specific targets. Relatively even levels of carrot red leaf, relatively even levels of carrot mottle. So it doesn't take a statistician to tell you that probably these 4 viruses that we're looking at aren't implicated in the symptom that we're seeing. So where do you go from there? Well, we took a deep sequencing approach, as I said, and this was really targeting RNA-based viruses, so looking at other viruses. And as I said earlier, we took 12 necrotic carrots, 12 apparently healthy carrots, but they actually had a mixture of virus and symptomatic status, so there were some with red leaf, some with mottle. And we ran 2 MySeq runs, one for the necrotic, one for the non-necrotic, and that gave us 16 million sequence reads. So as you can imagine, that's probably the worst sequence jigsaw you've ever had to put together. We've actually managed to script a lot of our bioinformatic work, so it's not quite as bad as sitting down with lots of A's, C's, G's and T's and putting those sequences together. And we then managed to look at that for the presence of both known and as yet unknown viruses. So what came out of that work? This data is presented in reads per kilobase of viral genome per million sequenced reads. Think of it as relative abundance, and then we don't get hung up on numbers. But as you'll see here, this virus, carotte yellowleaf virus, was far more prevalent in our affected sample than our unaffected sample. A carotte clostrovirus, this is a previously unknown virus, which is actually closely related to carotte yellowleaf, and that's present in more affected than unaffected, but by no means as common as a carotte yellowleaf. So it was unexpected that we found carotte yellowleaf. This virus has never been seen in the UK in that form. I think it was first described in Japan, been described in Holland, and we've now found it in the UK. But as I say, it's never been really looked for to any kind of depth in the UK. We don't know what the prevalence is, and this was almost an incidental finding. Alongside these unexpected viruses, as I say, we found beet western yellow's associated RNA. We think that's been tagged along with the carotte redleaf virus in the same way that the carotte redleaf associated virus is carried along by being encapsulated with the carotte redleaf. However, we also found some novel viruses, so these are previously undescribed. And within that, as I say, the carotte clostrovirus, which is in the same genus as carotte yellowleaf virus. We found 2 novel members of the beta flexiviridae, which we've tentatively named the cordoviruses. And the interesting thing is, whenever we do this sequencing, you occasionally get these beta flexiviruses popping up. So we think they're an incidental finding. We think they float around. We don't really think there's anything pathogenic going on there. The really interesting one for us was this finding of carotte terradovirus. There have been 5 terradoviruses described. 4 of those are in tomato. There's recently been one described in lettuce. All of the tomato terradoviruses are whitefly transmitted, and this is the first virus of this kind we found in the Apiaceae, first finding of its type. So obviously we wanted to investigate that further, but we also had to bear in mind the reason we went into this was to look at necrosis. So again, the carotte yellowleaf became our main target. So we went back and we designed specific real-time PCR assays to all of those novel and previously unidentified viruses so that we could go back and test our original field sample of 100 affected, 100 unaffected to see what the relative prevalence of those different viruses were. And as you'll see here with carotte yellowleaf virus, quite clearly nearly 96% of our affected carrots had carotte yellowleaf virus in them, as opposed to only 22% of our unaffected. And when we went back with all our viruses that we were expecting to see that we always test for, again, the numbers were relatively comparable, apart from you've seen that big drop in presumed negative because of all the carotte yellowleaf virus that we were finding in carrots. So at this point I felt obliged to hand it to a statistician. Not that I ever understand what they tell me, but he got quite excited at this point. And he designed us a way of analysing many different influences upon a system so that we could actually look to see what the association of either specific viruses or specific combinations of viruses were on necrosis. So just to explain this graph, the red line across here would mean that there's no effect on necrosis if you remove that virus from the system we're looking at. So the only time you see anything above this line really is where you've got carotte yellowleaf virus. So here, CYLV itself, any positive, and here you've got a combination between carotte yellowleaf and carotte redleaf. So that, to us, quite clearly indicated that carotte yellowleaf probably had some role to play in internal necrosis, and when we looked at the influence actually on the numbers, the influence of removing carrot yellowleaf virus from this necrotic carrot system was estimated to reduce the level of necrosis by 96%. So we feel that is the causal agent. However, we appreciate correlation is not causation. And that leads me on to the further work. So everything you've seen up until now, we've just had word that has been accepted for publication in PLOS One, so it'll be appearing on a website near you within the next month. We wanted to investigate further carrot yellowleaf virus. Obviously, Koch's postulates is a key part of that work. However, the only person who's ever been able to transmit carrot yellowleaf virus is Luc Bos, who was working out of the Netherlands in the 80s. He unfortunately died about 5 years ago, so we can't ask him how he did it. But we are trying mechanical transmission, we're trying aphid transmission, and all the little tricks that virologists have just to try and get this virus into alternate hosts so we can try and demonstrate Koch's postulates. We've been trying for 2 years and we're still struggling, so that's an interesting one to chase up at some point in the future. We want to look at transmission. We want to confirm that we suspect that the willow carrot aphid is the main transmitter, but we really want to look at that in depth and see what other aphids can transmit it. And what I'll talk about next is incidence and hosts of carrot yellowleaf virus, and just a small bit of initial work that we've been doing. As I said, because carrot virus is a really novel virus, and first time this group of viruses has been found in an apiaceous crop, we wanted to look at that as well. So we're trying to characterise that at the moment, and we also want to look at the field incidence, how much is actually out there. So as part of that work, we've now got a PhD student working with us who's doing a virome study, looking at the complete viral load within a given crop system, and she started with carrot and apiaceous weeds that surround carrot crops as our starting point. So to do that, this is partly funded by the HDC and also partly funded through DEFRA plant health development money. We're looking at wild hosts, so samples from umbelliferous weeds, or apiaceous weeds I should say, around carrot crops, and we're sampling those throughout the season and seeing which of these viruses are present. And we have found in one field up to 25% of cow parsley, Anthriscus sylvestris, within one field is actually infected with carrot yellowleaf virus. So that's giving us a good indication that this is possibly a weed into crop system that we're looking at. We also want to look at samples from carrot crops to see what the incidence is actually within those fields, and to see what the pattern of transmission is within those fields. Try and give us a feeling of whether some of these viruses may be coming in within seed, or whether they are insect transmitted issues. So as part of the PhD project, Zuri, our PhD student, is taking a 100 metre by 100 metre block within a field, taking a sample every 10 metres, regardless of whether that is symptomatic or asymptomatic, and then we're assessing foliar symptom development, and then we're going back at the end of the season, repeating the sampling exercise, but pulling roots up as well, to look again for what viruses are present, and also whether there's necrosis present. So just a few of our chamber of horrors that you might see. Again, it's not easy to show virus symptoms on a screen, but we think this is probably an individual virus, so either carrot yellowleaf virus or carrot torado. There's no clear correlation between a single virus and a virus symptom at the moment. But you get this very yellow leaf with quite clear green veining up the middle. What you also get are these very standout yellow leaves that when you walk into a field, you can quite clearly see them. And it looks quite similar to the pictures we saw yesterday of the Liberobacter symptoms. However, this appears to be a combination of viruses together. So you always find dual infections when you find those symptoms. So from one field we've looked at, it just happens that this field is about a mile away from my research institute, so we've been able to do quite a lot of work in this field. Again, given that the two viruses we're looking for here, carrot yellowleaf virus and carrot torado virus, up until 2 years ago we weren't even looking for within carrot fields. Carrot torado virus we didn't even know existed. And as you can see in this field, you've got carrot yellowleaf virus at 35% incidence. Each of these squares represents a single plant from those 10-metre sampling points. So you can see there's no real pattern to the transmission there. The carrot torado virus is at a lower incidence, but again, no real pattern to the transmission, nothing we can draw. But you've also got quite a few dual infected plants as well. So the total virus load on that field, just looking at these two viruses, is actually almost 50% virus. Given that a virus will hijack the plant's internal reproductive system for its own affairs, if that's not having an effect on yield, I'd be very surprised. But we've actually got to narrow down what the specific symptoms are. This is another field we've looked at, and I think this one's interesting because you've got relatively even levels of the yellow leaf virus and the Tornado virus. But if you look at the pattern of transmission, and again we've got to do a lot more work on this, you can see there's almost a diagonal line where you've got field margins and hedgerows to one side. And there's certainly more infection in that quadrant than there is in the other part of the plot. And that, to me, would indicate, given the similar patterns of transmission that we're seeing there, that carrot yellowleaf virus and carrot Tornado virus are probably transmitted in a very similar fashion by similar vectors. And so, given that most of the Tornado viruses we've looked for so far, in terms of the tomato ones, are whitefly transmitted, yet we don't really recognise whitefly as being a problem within carrot crops, I think we will concentrate on aphids in terms of where we look in terms of a vector for carrot Tornado. But as you can see, again, for viruses that we didn't even know existed 2 years ago, very high incidence within this field. So in summary, internal browning has been strongly associated with carrot yellowleaf virus, though further work is needed to actually biologically confirm that. Within that work, circa 12% of carrot yellowleaf virus affected carrots are expected to develop necrosis. So if you take those figures that we found this year, you could be looking at around 4% necrosis in certainly the more heavily affected field that we've looked at. But carrot Tornado virus has been found infecting up to 25% of carrots in surveyed fields, and we still don't know what's vectoring it or what symptoms it's causing. So I'd just like to acknowledge the HDC for their funding of the original carrot project, DEFRA, the UK Government Department for Environment, Food and Rural Affairs, for supporting the PhD studentship. I'd like to thank Howard Hines for not giving up on this being a virus-related problem. I'd like to thank Roger Hobson of Hobson Farming for allowing us into his fields quite readily. And I'd also like to thank the staff at the Ferrovirology, Molecular Technology Unit and Applied Genomics team for all the work they've put in on this. Thank you.
The roots that show the internal necrosis, do they exhibit any symptoms in the leaves?
That's what we're trying to find out at the moment. Because the work we did previously started on the processing line, we didn't have any foliage to go with. So we were going purely from necrotic root and working backwards. So the work we're doing this year is following crops through, so we can actually look at the foliar symptom and then come back and look at the root development later. So that will be finished within the next month or so.
Yeah, because for the analysis whether the virus is present in those plants, you used root samples.
Yeah.
Am I correct? So you have not made an attempt to demonstrate whether the virus is present in the foliage.
In terms of this work, no, we haven't. No. Because again, we wanted to start from the point where we were seeing the problem. But we've had samples sent in where you have root necrosis and symptomatic foliage, and you find the virus in both.
Okay, thank you.
Hi. Do you have any idea why only 12% of the plant will develop necrosis?
That's a good question. I think there's something to do with timing of infection. There's probably something to do with local environmental conditions. But until we get further down the line of these projects and actually try and work out that causal relationship a bit more closely, one of the aspects of the work we're doing on Koch's postulates is actually to look at the delay from infection to symptom development, to try and work out whether you get kind of a first flush of aphids coming through a crop that will bring the virus in, and then if you get sequential transmission within the crop from there that happens later in the season, does that mean the first ones that get hit will get the necrosis and then later on they won't? So it's an unknown quantity, and we're postulating at the moment, I think.
Okay, one more question.